Hello, everyone, and welcome to Article Advances, an interview series where we take a closer look at publications related to comprehensive genomic profiling, also known as CGP, and cancer. My name is Jamie King, and I'm a senior medical writer at Foundation Medicine. In this video, we will hear from Rachel Evans, a translational scientist at Foundation Medicine and an author on a recent publication featuring an analysis comparing methods of fusion detection, exploring why breadth versus depth matters. So, Rachel, we know that gene fusions are a type of genomic alteration that can drive cancer development and progression. How are they relevant for clinical decision making?
So, gene fusions have clinical value for diagnosis, prognosis, and treatment since there are approved targeted therapies or evidence of utility for informing treatment response for specific gene fusions in specific cancer types, all of which can lead to improved outcomes for patients.
And how have advances in next generation sequencing or NGS technology impacted fusion detection for clinical utility?
So, whereas previous methods for fusion detection were limited to single gene loci, NGS technology allows for broad profiling for both known and novel fusions that may inform clinical decisions. So, the sensitivity, along with specific design features of NGS assays, are important considerations for fusion detection since many gene fusions are complex in nature.
Great. So, to start discussing this article, what was the rationale for comparing the two methods of fusion detection, RNA CGP versus whole transcriptome sequencing, also known as WTS?
So, as we've established, robust fusion detection is important for detecting clinically meaningful alterations, and we examined how both methodologies, WTS and RNA-CGP, perform for detecting actionable fusions, as these are two well-established but technically very different sequencing methods.
So, as we think about these two methods, can you first describe a benefit of WTS?
Yeah, so WTS covers a larger number of genes than RNA-CGP, offering wider breadth of coverage and allowing for potential discovery of previously uncharacterized alterations, some of which may not yet have a clinical impact.
And when we think about RNA CGP, can you describe a benefit of that method?
So, RNA-CGP offers a higher depth of coverage for improved sensitivity, allowing for detection of low abundance alterations. And this approach may enable more focused efforts for detecting clinically actionable fusions with confidence versus expansive sequencing that may yield additional information, but information that may not be clinically informative.
And so, how are these two methods compared in this study?
So, in the first analysis, we used a commercially available fusion positive control sample representing a panel of clinically relevant fusions to examine fusion detection with Foundation 1 RNA, a laboratory developed test for the detection of rearrangements and fusions. And we compared detection with the Foundation 1 RNA assay to published validation data for a WTS assay, which used the same fusion positive control sample. So, with this approach, we observed reduced sensitivity for detecting fusions with WTS versus RNA CGP.
And then another approach in this study utilized clinical samples to compare WTS and RNA CGP. How do the rates of fusion detection compare in this approach?
So, in the second analysis, fusion positive clinical samples were tested using RNA CGP, which was performed using either Foundation 1 RNA or another assay, Foundation 1 Heme, which also combines DNA and RNA sequencing. And we also tested these samples using Foundation Medicine's research use only WTS assay. And RNA-CGP detected 100% of fusions across 20 fusion-positive clinical samples. However, with WTS, 6 of 20, or 30% of fusions, were either not detected or were detected below the confidence thresholds for clinical reporting.
And so, in this study, are there limitations associated with either method?
Yeah, so generally, because the total number of sequenced reads per sample is limited by sequencing costs, sequencing reads are spread more thinly across genes when using WTS, which results in lower depth of coverage compared to RNA-CGP. With WTS, highly expressed genes, some of which may not be cancer associated, might consume a significant proportion of available sequencing reads, and fusions with low abundance in a clinical sample may be missed because they have to compete with these highly expressed genes for sequencing coverage.
And so, how did the findings here provide insights on missed fusion detections?
So, while the proportion of fusions not detected by WTS may be higher or lower than reported in our study and is dependent on both assay specific and sample specific factors, our findings demonstrate how the reduced sensitivity of a broader WTS approach could potentially lead to missed fusions.
And so, in thinking about future directions, what are some future directions that could stem from this analysis?
So, the sample size in this study was small and only allowed testing of a subset of clinically relevant fusions. So comparative studies using larger and more diverse clinical cohorts could help the field better understand how frequently fusions could be missed with WTS in real-world clinical practice.
Great. Thank you so much for your insights on this publication, Rachel. And thank you all of our viewers for your attention. We look forward to the next Article Advances feature.